The following presentation will be on the light reaction of photosynthesis. So this is the absorption spectrum of chlorophyll A. You can see it absorbs really well in the violet and blue, not so well in the green, and pretty good in the red area. Chlorophyll B is also a very common pigment in plants. It absorbs really well in blue, not so good in green, kind of good in orange. Okay, you'll notice though both chlorophyll A and B, so we got chlorophyll A and B, they both are very poor at absorbing in this region down here, that 500 to 550 region, and that's kind of the green area. So we don't absorb green very well. Matter of fact, we reflect. Whatever you don't absorb very well, you reflect. And so we reflect the green and some of the yellow colors off, and you look like what you reflect. And so we're absorbing this part of the spectrum, and we're absorbing this part of the spectrum, and we're reflecting this middle part of the spectrum, and so that's why plant leaves look green. They also have some accessory pigments in them, carotene and xanthophyll and some others that are orange and red and yellow and some other colors. These are usually not noticeable because they're found in lesser amounts. But in the fall, chlorophyll is pretty wimpy. And so when it starts getting cold at night, chlorophyll's cold sensitive and it starts to break down and gets resorbed by the plant. And this exposes the other pigments. So in the fall, we see the reds and the yellows and the oranges that were there all year. They just don't become visible until the fall. So you can see here the chlorophyll is breaking down, getting resorbed, and the other pigments are becoming visible. Eventually, the whole leaf will turn a certain color. Sometimes it'll go through phases. It'll go through yellow to orange to red. All those pigments are in there. They're just being resorbed by the plant at different rates. So if we look at a bunch of different pigments, you can see a lot of the accessory pigments are there to help absorb this region that plants don't absorb very well. Chlorophyll A and chlorophyll B don't absorb very well. Because these are not found in as high a numbers, they don't do as good a job of absorbing this region, but it kind of helps out. If we look at the overall absorption spectrum of photosynthesis, it mostly absorbs the violets and blues. It doesn't do a very good job with the greens. It gets a little bit of the yellows and oranges and reds, but it's better than what the chlorophyll by themselves was. The accessory pigments boost those areas that were not so good. All right, so how do we get the actual energy from this process of the light reaction? So photons are going to hit reaction centers, and they're going to hit these accessory pigments these pigments that are embedded in the thylakoid membrane, and the energy is going to bounce around from pigment to pigment to pigment until it gets to a reaction center. The reaction centers are little chlorophyll that are good at sending the energy from the electrons, energizing those electrons, and boosting up the primary electron acceptor. Okay, so we get these electrons energized and boost up to a high level so that they can be, the energy can be used from those electrons as they pass through the electron transport chain. The photosystem 2 and photosystem 1 have slightly different primary acceptors, but they basically work the same way. They do get their electrons from a different source. The electrons from photosystem 2 come from water. So we split water. The electrons are lost, which makes our hydrogens positively charged. And 2 water molecules will split. And then oxygen from each one of those water molecules will become part of O2. And then O2 is given off as a waste product. That's where we get our oxygen gas from. And the hydrogens then are used in a process where they build up and then they flow through the ATP synthase later on. We'll talk about that in a minute. So here is our kind of our whole system. Photosystem 2 comes before photosystem 1. Photosystem 1 was actually discovered first, and they named it photosystem 1 after they discovered photosystem 2. And then it was only later on that they figured out the sequence, and so they just left the names photosystem 1 and photosystem 2. But photosystem 2 actually comes first in the sequence. And so what happens is light gets absorbed, transferred to the reaction center, where the electrons get energized to a higher level. And then as they pass through an electron transport chain, hydrogens get pumped into the thylakoid space. So this is the stroma out here, and the hydrogens are getting pumped into the thylakoid space. As hydrogens get dropped off from water and hydrogens get pumped in, we get a buildup of hydrogens inside this thylakoid space. As they flow out, just like what we saw in cell respiration, you're going to have an enzyme called ATP synthase produce ATP from ADP and phosphates. So the energy from this flow, this chemosynthesis, chemoosmosis, causes the production of ATP. Those electrons that have kind of lost their energy get re-energized in photosystem 1. They then transfer to another system where they will help reduce NADP. It's done by an enzyme called NADP reductase. A lot of times enzymes will be named by the job that they do, and so that's the case here. And NADP enters and leaves as NADPH. So it's been reduced because it gained electrons and it gained a hydrogen. Okay, so we end up with NADPH there. And both NADPH and ATP are going to be used in the Calvin cycle to help produce sugar. And we'll get to that later on. Both of these processes that we see in the mitochondria and the chloroplast are very similar. They're kind of reversed. In the mitochondria, hydrogens are pumped out and then they flow back in. In the chloroplast, the hydrogens are pumped in and then they flow back out. But other than that, it's a very similar process that we see in both. Probably early on, these guys were related. We'll talk about a theory later on where we think that mitochondria and chloroplasts might have been independent organisms at one point. We're not 100% sure, but we're pretty confident that that's the case.